Let's take a look at chapter 3 in your textbook. This chapter is all about functional compartments or body fluid compartments. So we're going to take a look at the functional compartments. Biological membranes is a big part of it because these are the borders of these compartments. We're going to also take a look at some intracellular compartments, so that would be within the cell, additional compartments, and then tissues briefly, tissue remodeling also very briefly, and then a little bit about organs. So let's take a look at the functional compartments. In physiology, it is really important to distinguish between the extracellular fluid compartment and the intracellular fluid compartment. Let's take a look at this image right here. It makes it maybe a little bit more clear. So the extracellular fluid compartment, abbreviated as ECF, that would be the fluid outside of the cells, and that is roughly 1/3 of the body fluid, total body fluid. And the fluids outside of cells would be what's between the cells, and that's the interstitial fluid, and then in your blood, what's traveling in your cardiovascular system, so that would be blood here. The fluid that's inside of cells is known as the intracellular fluid compartment, the ICF, and that is about 2/3 of your total body fluid. And well, you can see here a variety of cells, so all the fluid inside of cells. So that is still twice as much volume as what you have between the cells. And now we're going to take a look at the borders. So it's very important to consider the borders between the compartments. And there you have a plasma membrane right here. So here's a cell, this cell right here, and the cell has fluid inside of it and it has fluid outside of it. Okay, and the border that's keeping the two compartments separate is called a phospholipid bilayer. Phospholipid bilayers are very important in physiology, as you will see. We will talk a lot about that. So phospholipid bilayers, as we saw in a chemistry lecture, they are phospholipids. They have this phosphohead group and these two fatty acid tails. And take a look at how the two fatty acid tails, they're facing one another. So you have this nice lipid barrier in the center right here. And then the phosphohead groups, they actually like to mix with water. So they're more hydrophilic. And so therefore they make a nice junction to the water-based exterior and the water-based interior of the cell. And then as a nice border, we have this lipid layer that is not permeable to water, is hydrophobic in nature. So the cell membrane function is to physically separate the two fluid compartments, the extracellular and intracellular fluid compartments. Now, you don't want just to have a barrier. It's just like in your house. You do want to have your walls, but you also want to have windows and doors so you can get stuff in and out. Including yourself, of course. So, but in cells, the same thing. You need to have exchange with the environment. You want to get rid of your trash. You want to pick up nutrients. You want to get the good stuff in and the bad stuff out. And you need to communicate between cells as well. So, the cell membrane has a variety of functions. It regulates the exchange of materials. And then it also helps with the communication between cells and the environment. And then they offer some structural support because your cytoskeleton is linked to the cell membrane. So we can take a look at sort of some profiles of a variety of membranes. They are pretty much all phospholipids embedded with a variety of proteins. So you have various amount of lipid depending on how much protein is stuck in that membrane for communication. So if you take a look at a red blood cell membrane, you will find there's a lot of protein, but still you have quite a bit of lipid as well. The inner mitochondria membrane, on the other hand, here has actually even more protein. There's so many protein molecules, signaling molecules, and for the electron transport chain, these proteins are stuck in that phospholipid bilayer that now the protein is even the majority of what the phospholipid bilayer there is. But anyway, so here let's take a look at the composition of cell membranes. Of course, the major component are these phospholipids, so these lipids. And we have a variety of phospholipids. They come in sort of different flavors. it's not that important for you guys. Sphingolipids are in there too, and there's a significant amount of cholesterol present in biological membranes, in animal membranes, that is. And then, as you just saw, there can be a very significant amount of protein stuck in that phospholipid bilayer. So some cells have more protein than phospholipids in their membrane. But anyway, so the proteins, they can be completely embedded into the phospholipid bilayer. Then we call them integral membrane proteins. And so there will be transmembrane proteins or lipid ingot ones. And then we have peripheral proteins that are just sort of attached to the membrane, but they don't span the entire phospholipid bilayer. We also have some carbohydrates such as glycoproteins and glycolipids that can be attached to the phospholipid bilayer. But here's a simple diagram of just the phospholipid bilayer. It's just this arrangement of these phospholipids where you have the polar head group and a non-polar fatty acid portion, two fatty acid tails there, and tan is tan color. They like to arrange themselves so that the fatty acid tails are facing one another. So you have two rows of phospholipids here, and then the hydrophilic head groups, they're facing outside to make a nice juncture to the water-based exterior and interior of the cell. And so here is a very simplified model. You can actually generate these little phospholipid spheres in the lab. It's not even that hard. So these phospholipids, they like to arrange themselves in this manner automatically. Now in biological membranes, you have a bunch of stuff that's stuck in that phospholipid bilayer. Let's take a look at first this yellow stuff that's stuck here between these phospholipid molecules. That's cholesterol. And biological membranes have a significant amount of cholesterol as part of the membrane. The cholesterol adds what we call fluidity to the whole system. So the whole thing is liquid, and then the cholesterol adds additional liquidity or fluidity to it. Nothing is static about biological membranes. They're all very dynamic. And you can see here a bunch of integral membrane proteins. So here's a transmembrane protein. It's completely spans the entire membrane, and it's linked to here some cytoskeletal peripheral proteins. And then here, this would be a glycoprotein. You have the carbohydrate residual sticking up. So over here, we're going to have a membrane protein that has several transmembrane domains. So these ribbons here that go all the way through the phospholipid bilayer, those are transmembrane domains. And proteins can have various number of transmembrane domains. One membrane protein that we'll talk a lot about is called the G protein coupled receptor. And that one has 7 transmembrane domains. So 7 of these ribbons that are, of course, these are alpha helices from the protein. These are secondary structures of these proteins, and they will span the phospholipid bilayer, and some of them they can actually they can form pores then that allow the transport of different materials. Okay. So here's a diagram basically telling you the same information that I just talked about. And here you have the cell membrane composed of cholesterol, then you have these phospholipids, and then a bunch of protein. Protein is very important, also carbohydrates sometimes. And if the carbohydrate is attached to a protein, then we call it a glycoprotein. If it's attached to a lipid, it's a glycolipid. And again, the main structure of the biological membranes is this phospholipid bilayer, which has also a significant amount of cholesterol. Okay, this is what this looks like in an electron micrograph. It's kind of cool. It gives you a little bit better idea of what this is really like. In blue here, you have the cholesterol embedded there, and then you have these what we call lipid rafts, or a cluster of proteins that have sort of travel together. Maybe they're signaling molecules, or it's a cluster of membrane proteins that kind of travel together and stay together. So picture a raft on sort of the ocean of phospholipids, I guess, in this case. And here, moving on, So we just talked about the cell membrane. That's the border of the cell. So the cell membrane is out here, the border that separates extra and intracellular fluid compartments. Inside of the cell, you're going to have a variety of different items. The nucleus is the most obvious, biggest extra compartment within the cell, and that one contains the genetic material. We'll talk some more about the nucleus. The cytoplasm is all the stuff between the nucleus and the cell membrane, and there you have a variety of organelles. We will talk about mitochondria. They are responsible for your energy metabolism. The endoplasmic reticulum, Golgi apparatus, they are all involved with protein synthesis. Then we have lysosomes and peroxisomes. That's sort of the cleanup crew of your cell. And there are some inclusion bodies that we will not spend too much time on, except for ribosomes, which are also, of course, involved in protein synthesis. And then we have protein fibers that are for the cytoskeleton. They also form centrioles and cilia and flagella on the outside of cells. So intracellular compartments, we call that cytoplasm, and there we have all these different items: the cytosol, inclusion bodies, the cytoskeleton, these fibers, and then the organelles that are in there, and plus the biggest one, the nucleus. Okay, inclusion bodies, they don't have any membranes. The one that we will be talking a lot about, the only one we will talk about, is the ribosome, and that one is involved in protein synthesis. That's the part where protein synthesis actually happens. Most ribosomes, they are fixed. They're sitting on top of the endoplasmic reticulum, making it the rough ER. And we have some free ones as well. They can also form polyribosomes, but we will talk more about the ribosomes when we go do protein synthesis. Here is this lovely picture. It's not very good, to be honest, but it's in your book. And it does show the endoplasmic reticulum is sort of this endomembrane system in purple here. And you see the little dots on top. Those would be, those are ribosomes, fixed ribosomes that sit on top of the ER, making it rough ER. And then let's continue here with some protein fibers. The most prevalent fiber that you find in cells is definitely actin. Those are microfilaments. So we also have medium-sized ones, intermediate filaments. A lot of them are keratin fibers, keratin filaments, neurofilaments, for example. And then the largest ones are microtubules, and they form cilia and flagella along with centrioles when the cell is dividing. So here is sort of an overview over the fibers that you find in cells. Are 3 different sizes. The smallest ones are microfilaments, and then intermediate fibers, intermediate in size, you can see that 10 nanometers. That would be these keratin fibers, and then microtubules, the largest ones, 25 nanometers. And here's some examples. Tubulin is a good example of microtubules. So let's take a look at the centrosome, or some microtubule function. Centrosome for assembling of tubulin monomers. Centrioles are important for cell division, and cilia, flagella, those are for movement outside of cells. And here, not such a great picture again, sorry about that, but it's in your book of centrioles, so I can take a look at that. Here are some examples. You can see here cilia and flagella. These are cell extensions on the I shouldn't say extensions, but they are attached to the outside of the cell. Flagella very long and help the movement of the cells. There's only one human cell that has a flagellum, and that would be a sperm cell. Several cells have cilia, like in your respiratory tract, you have ciliated epithelium, and the cilia and flagella, they are all composed of these microtubules, and you can take a look at that on this image, but you don't need to know the details of this. The cytoskeleton function is about the same as in your bones for your skeletal system. Gives the cell structural support, and so nothing in the cell is random, even though it may seem like that sometimes, but the cytoskeleton is sort of the infrastructure of the cell and gives the cell its shape and makes sure that things can be transported to where they need to go. Okay, A little bit more here on the cytoskeleton. You can see how the cytoskeleton, these for example, they can form microvilli on the top of here of an absorptive epithelium, like in your small intestine. You need to form these microvilli. These are true cell extensions, so they increase the surface area, and you will do that with microfilaments that are forming then this structural network to have microvilli. We also have motor proteins. You'll hear much more about them when we do muscle physiology. But here are some examples of motor proteins, and they, under expenditure of energy, they will transport stuff. We have myosins, and that's a personal favorite. I'll tell you why when we get to the muscle physiology. That's one motor protein that you need for muscle contraction. And then we have also kinesins, dyneins. They can move vesicles along microtubules. They're pretty cool. So take a look at this. I'll post probably a little video showing you about cell structure, so the inner workings of the cell. There's a pretty cool video out there on YouTube. Here's an organelle that's being transported along a cytoskeletal fiber. So here is your infrastructure, sort of the road on which this motor protein can walk, and it's using literally its little feet, moving 1 foot ahead of the next, transporting something in this direction. Okay. Moving on to mitochondria. Think of mitochondria as the power plant of the cell. This is where you're creating ATP, so cellular ATP production. That's what's important about mitochondria. And we will talk more about them when we do the aerobic respiration lecture. So here is a not super great, but okay picture, an image of a mitochondrium. You can see there's an outer smooth membrane and an inner mitochondrial membrane. So there's a two membrane organelle, and that's very important, as you will see when we talk about aerobic respiration. Moving on to the endoplasmic reticulum. It's sort of an endomembrane system that's involved in mostly with protein synthesis, but also with some fat metabolism. A rough ER has ribosomes on the outside, and it's why it's rough. And there you have protein synthesis and assembly and modification. Smooth ER doesn't have any ribosomes, and so there we have more lipid metabolism that takes place there. Synthesis of fatty acids, steroids, and lipids. So here, again, one of those not so creative images that you'll find in a textbook, but good enough. Rough ER has the ribosomes outside of it, and that's why it looks rough. And then smooth ER, lipid metabolism, no ribosomes. The Golgi apparatus is sort of the final step in protein synthesis, sort of your packaging and distribution center. It's also an endomembrane system, stacked membranes basically surrounded by vesicles. It takes the almost finished product from protein synthesis from the endoplasmic reticulum, and then packages it basically does final steps, final attachments, and then we will package our protein products into vesicles and ship it. So the Golgi apparatus attaches sort of a shipping label and tells the cell where this protein needs to go. And here, amazing, another image that's not super great, but you get the idea. The Golgi apparatus, sometimes it's referred to as, it looks like a stack of pancakes. Here they color-coded it in purple, but anyway, so you can see little vesicles that are fusing and pinching off the Golgi apparatus. So some more cytoplasmic vesicles. We also have secretory vesicles, storage vesicles, and then lysosomes and peroxisomes. And these are more the cleanup crew of the cell. They degrade bacteria, or they recycle old organelles. And then peroxisomes, they are involved with some hydrogen peroxide generation and making sure that the hydrogen peroxide doesn't do any damage to the cell. Okay, so here now, this is the ultimate creative image of lysosomes, these little green dots. I'm kidding, right? You know that. It's not a very good image. I can't I don't know, the person who made this slide or this image, created this image for the textbook, was definitely not very creative. And you can see that here, 2 peroxisomes. The only thing they changed from the lysosomes to peroxisomes, I feel like, is the color. So they color coded these in purple. But anyway, moving on to the nucleus, and that is the control center of the cell. This is where the genetic material is localized. And we're protecting the nuclear material with 2 membranes. So we have a double membrane bound organelle right here in the nucleus. We do have a nuclear pore complex that allows for transport of stuff in and out of the nucleus. We need to have a fairly complex nuclear pore complex because a large molecule like messenger RNA, for example, that was made in the nucleus needs to be able to get out, and not so simple. Chromatin is the term that we use when we say it's DNA plus associated proteins. So there's a lot of histone proteins in the nucleus that help organize the DNA. Together, the proteins plus the DNA make chromatin. And then we have nucleoli. These are referred to as little nucleus within the nucleus, and they are the site of RNA synthesis. And this is mostly where we make the ribosomes. So here's a picture of the nucleus. They got a little bit more creative on that end here. You can see the nuclear pore complexes. Those are little dots on the outside. Inside, you can see there's sort of this chromatin material, protein plus DNA. And then we have also the little nucleus, the nucleolus, which is the site of ribosome manufacturing. Here's a pretty elaborate slide that shows you sort of the flow of the information from DNA to finished protein product. And you can see how many of the organelles that we just talked about are involved with that. So here we start out with transcription, which results in messenger RNA, and then the messenger RNA needs to be translated to protein using ribosomes. All of that costs you a lot of energy, so you need mitochondria to churn out a bunch of ATP as well. And then these almost finished, or these sort of raw material of proteins, they need to be modified, and that's the job of the ER. So this is in purple right here, this endoplasmic reticulum. And then they hand off the almost finished product to the Golgi apparatus, which makes final modifications, and then we'll either dump the contents outside of the cell or ship it within the cell wherever it needs to go. So you can read through the steps here and follow along with that. I did this kind of fast.